Hello, this is Kangjin Kim from Korea, and let me talk about the role of unclassified Lachnospiraceae in the pathogenesis of type 2 diabetes. First of all, let me talk about urine microbiome data from extracellular vesicles, EVs. EVs has been recently suggested to be the main messengers between intestinal microorganisms and the host. In other words, microorganisms inside of gut can affect entire body with their EVs. And inside of those EVs, there are EV components, and these include nucleic acid, which we can analyze with metagenomic analysis. However, microbiota-derived EVs are variable because they are affected by various factors, and this makes it hard to infer causal relationship. And longitudinal microbiota study can allow stronger inferences than cross-sectional studies. And this is why we investigated the prospective Korean Association Resource Project, CARE cohort. This is longitudinal microbiota research, and it includes multi-omics dataset. With this CARE core study data, we conducted 2-on-1 propensity score matching with age, sex, and BMI. As a result, we have 328 healthy subjects, 164 T2D at-risk subjects, and 199 T2D subjects. After pre-processing, we have 393 individuals with 3 times repetition. CARE core study includes metagenomic, functional profile, clinical, metabolite, and genetic data. With these multi-omics data, we conducted association analysis with type 2 diabetes and network analysis with target microorganism and conducted Mendelian randomization analysis. This plot describes the overall profiles of metagenomic dataset, and it says relative abundance of some genera changes over time. For example, Bacteroidetes and Akkermansia went down during the follow-up periods. The result of Permanova says the top 4 variables significantly explained the amount of variance of microbial change for 4 years. And this includes HbA1c, which is the main indicator of type 2 diabetes. For the association analysis of T2D with bacteria genera, we dichotomized T2D groups. T2D at risk / T2D means the subjects are in T2D at risk group or T2D group. Binary T2D means the subjects are in T2D group. And the result of the association analysis says that GU174097 was negatively correlated with the risk of type 2 diabetes after multiple testing correction. For the association analysis of T2D with bacteria genera, we dichotomized T2D groups. T2D at risk / T2D indicate 1 when the subject are in T2D at risk group or T2D group. Binary T2D indicate 1 when the subject in T2D group. So, for the association analysis result, GU174097 was negatively correlated with the risk of T2D after multiple testing correction. And this GU174097 was an unclassified Lachnospiraceae. For detailed visualization, we divided the subjects into 4 different groups. The first group consists of subjects who had type 2 diabetes in all the 3 phases. The second group has subjects who were healthy in the first phase but became type 2 diabetes patient or T2D at risk group in phase 3. The third group consists of subjects who were T2D at risk or T2D in first phase and healthy in third phase. And fourth group consists of subjects who were healthy in the all phases. In this plot, group 1, 2, 3, 4 that I mentioned before has their corresponding color, red, green, blue, purple. And let's focus on green boxes. Green box means the subjects were healthy on the first phase but became T2D or T2D risk group on the third phase. And we can see that the relative abundance of GU174097 went down as time changes. And this means the target microorganism is not only related to type 2 diabetes status, but related to the progression of type 2 diabetes. When we use metabolite data, we found out that GU174097 was negatively correlated with ketone bodies. In this plot, we can see that the abundance of ketone bodies can also be correlated to the progression of type 2 diabetes. The network says that GU174097 was positively correlated with 60-minute insulin and negatively correlated with ketone bodies. And these ketone bodies was positively correlated with 60-minute glucose and 120 minutes glucose. After that, we used genetic information to investigate the causal relationship. So we used Mendelian randomization and utilized associated SNPs as instrument variables. So we considered these 5 different MR methods, and we conducted these 3 different GWASs and found associated SNPs. We conducted extensive assumption checks for 2-sample Mendelian analysis and found appropriate MR method for each combination. These are the causal estimate and the p-values for each combination of exposure phenotype and different MR methods. The significant result says that acetoacetate can increase HbA1c. The FDR corrected p-value was 0.046 for MR ag-Cmex method. This plot shows overall information of instrument variables and the result of each ML methods. The result of longitudinal association analysis showed that GU174097 was associated to the progression of type 2 diabetes. Also, network analysis showed that GU174097 was correlated to insulin level, and it was also correlated to ketone bodies. MR results showed that acetoacetate can increase HbA1c. And by the previous researches, a member of Lachnospiraceae, Coprococcus uses a member of ketone bodies to produce short-chain fatty acid. And this short-chain fatty acid can protect against type 2 diabetes. So in summary, G174097 was negatively correlated to the progression of type 2 diabetes, and the exact mechanism is unclear. But we saw a connection with ketone bodies, and by the result of MR method, this ketone body can increase HbA1c, which is a direct indicator of type 2 diabetes. And we found a connection with the previous research that Lachnospiraceae can use ketone bodies as energy source and can produce short-chain fatty acid and can lower risk of type 2 diabetes. But this study has limitations. The replication as MR is hard to be conducted because G174097 is a unclassified microbiome. So we lack of detailed information, and we lack of the result of genetic study with urine EV microbiome data. And since our data is repeatedly measured, there can be some kind of discordant result with the cross-sectional dataset. And the exact mechanism how G174097 and acetoacetate can affect type 2 diabetes remained unclear and need further investigations and experiments. This is all I have for this presentation. Thank you so much.